All right, good afternoon. Welcome to the Public TIC IPM Working Group July meeting. If you're new, welcome. If you're returning, welcome back. Today, we are very excited to have Dr. Roger Laine from Louisiana State University. He teaches in the Department of Biological Sciences, and today he will be discussing Nootkatones as a safe new class of natural insect repellents with a large market potential, and their chemical engineering for economic synthesis. This program is funded by the North Central IPM Center, supported by the USDA National Institute of Food and Agriculture, Crop Protection and Pest Management Program. So thank you for all you do. This would not be possible without your support. The TIC Academy, sounding like a broken record here, but save the date, please, October 16th and 17th. Registration will be open, really hoping for next week at the latest. If you're interested in a sponsorship opportunity, please email me. My email is right here at the bottom. And if you have recommendations for speakers or topics, please email me as well. So look for Basecamp announcements about registration very soon. In August, we are going to skip the August webinar, so our next webinar will be September 11th at 1 p.m. Central Time, with a speaker to be announced in the near future here. Once again, if you're interested in presenting for a webinar or have information to share with the group, please email me and we'll get you all set up here. Any updates from working group members? Anything fun going on? Working on something new? Anything like that? If anyone thinks of anything, feel free to put it in the chat throughout the webinar or email me later on and I could put it out to the group. So today's speaker, Dr. Roger Laine, is a professor of biochemistry and chemistry at the Louisiana State University Department of Biological Sciences. Dr. Laine has been involved in Nootkatone research since it was discovered as an insect repellent by a member of his laboratory, Betty Zhu. He is the sole author of the most recent patent for the scheme of organic synthesis of Nootkatone, which is being scaled up by Professors Dooley and Benson in the chemical engineering department. They have a joint NSF grant for $550,000 over 2 years for the chemical engineering scale up. And today he's going to be discussing some history and giving all kinds of information about the product and the work that they do over there. So while he gets set up, I'm going to launch our poll question. This will be a true-false format. So I'm going to stop sharing my screen. And everyone, please welcome Dr. Laine. You can go ahead and share your screen while they finish answering the poll.
Let me know when you're ready.
Go ahead.
Okay, so Nootkatones we discovered in our lab rather accidentally, I would say. And most of the stuff that's happened that's been fun in research to me has been an accident in the lab where we found something we didn't expect. Dr. Carrie Dooley, a professor of chemical engineering, and I submitted a grant application to the Department of Defense in 2022. It almost got funded in a program because they're interested in tick.
Sorry, Dr. Laine, can you share your screen?
Sorry?
Can you share your screen? We can't see your slides.
You can't, am I muted?
No, we can hear you, but we can't see your presentation.
Oh. I thought you had it.
Should be able to hit the green share screen button. Just perfect. Yep, there it is.
You got it?
Sorry about that. Yep, we're all good to go. Thank you.
Okay, so we had been working on this for a number of years and had real trouble with trying to get a scale up synthesis because Nootkatone is too expensive to use for an insect repellent. And I'm going to go through all that. So we applied to the Department of Defense. They had tick-borne diseases effort. We almost got funded, and we sent the same grant in 2023 to the NSF. And December, they gave us indication it was going to be funded, and in March we got funded. So we're 2 years in a chemical engineering to try to scale this up to the kilogram level. Now, if I can get my slides to change. Okay, the purpose of my talk is just to discuss the uses and markets for Nootkatones to reduce the cost of Nootkatones for insect repellent use, in addition to its use in flavors and fragrances. Describe the new synthetic method. We're engaging chemical engineering, and we're looking for partnerships for licensing agreements. Nootkatone is a major flavorant of grapefruit. If you opened a little vial of this, the whole room would smell like grapefruit. So it's the principal fragrance, and it's used in the fruit juice and confection flavoring industry, where it's regarded as safe. So doesn't have any regulatory problems for ingestion. And it's also used in the fragrance industry and perfumes, so it doesn't have any regulatory problems for topical use. It's an aromophenylene sesquiterpene ketone. That's the generic name for it. And it was first isolated and identified from the Alaskan yellow cedar tree, but it was not identified as an insect repellent. It was just identified as a terpene in 1962, but that's a highly insect resistant tree. In my lab, we had an incident where a fellow who owned a greenhouse in New Orleans came to talk to us. He had vetiver grass growing in, and he never had an insect problem with the vetiver grass, and he wondered why. And so we looked it up, and it turns out that the people in India had been working with vetiver for ages, and they knew it had insect repellent properties. They used to take roots and put them in a little cloth bag and then put them in the drawers with wool or silk. And the Indian chemists have identified 300 compounds in vetiver oil, and the principal component in there is something called vetivone. I'll show you the structure of that later. But the reason we told this guy is because 50% of the oil in that thing is a compound called vetivone, which has weak insect repellent properties. Then I asked my long-term colleague Betty Zhu, who's from the Shanghai Institute of Biochemistry and has immigrated. I asked her to look in the Fisher Sigma catalog because they have pictures of compounds in there. And I asked her to look for any compounds that had a similar structure to vetivone. And so she found 25 compounds, and one of them was Nootkatone. And we tested them in a termite repellency test to start with, and Nootkatone was the most potent one. So that's how it was discovered, just by looking through trying to find something that resembled a weak insect repellent. So we've had a number of publications and patents on this. We tested it on mosquitoes, ticks, fleas, tested by Otsuka on a mouse model. Lice I tested on my own son, caught it in school about 20 years ago when he was 8 years old. And other insect pests that we've tried are termites for sure. cockroaches and fire ants. I had recently a lot of people working on fire ants, undergrads in my lab. So here's the structure. And so this is just a chemical name for it, which you can see is 3 lines long. And it's a double ring terpene with a ketone group. Properties are melting point 32. So put it in the refrigerator. It crystallizes. And some of this work had been done by Badukian, which is a flavor company in Cincinnati. So here's the way the thing is synthesized. First, I'll go through this once here, and I'll review it later. But the way it's synthesized in the world is it starts with balancine, which is from orange oil, which costs $700 a kilogram, and a kibbutz in Israel called Aramor does one oxidation step and then cleans it up and sells it for $2,500 to $3,000 a kilogram. The compound that I first talked about, Vedivone, is in the upper right-hand corner here. And you can see that the only difference in these compounds is the location of the double bond in the right, in the little isopropanyl group. And it makes a huge difference in the scent. So Vedivone is kind of musty, dusty, and Nootkatone is grapefruit. So our sensors, our gustatory sensors and so forth, are really sensitive to structure. So we reduced the ketone group to Nootkatone, and there was no activity. So I put some numbers here of insect repellency. So we'll call Nootkatone 1, balancine 0, Vedivone is half as effective, and Nootkatone has no effect. So that ketone group was necessary. Then we reduced the double bond in the isopropanyl group, and it's called 11,12-dihydronootkatone, and that had the same activity as Nootkatone. Then we reduced the double bond in the ring, which you can do selectively, and that's 1,10-dihydronootkatone, and that's 4 times as effective as a repellent on termites than Nootkatone. And when we reduced both double bonds, it was 10 times better. That's tetrahydronootkatone. Those are also sold as flavors and fragrances at a huge price, as you can see. Now, the market's quite large for Nootkatone. It's 1 metric ton a year, so that's $50 million, but it's at that price. So acute toxicity, this was done in 1977. Oral studies in rats exceeded 5 grams per kilogram, so it's very, very non-toxic. And very small amounts give you a lot of flavor. Dermal studies were done, rabbits, and also the LD50 exceeded 5 grams per kilogram. And skin irritation was done on humans, and no irritation was observed after 48 hours. Closed patch test with 10% in petrolatum on the backs or forearms of people. And so animal studies were also done. There were rabbit studies done for skin irritation. Also, there's no skin sensitization. So the tests were done by a couple of different labs, and they are trying to see whether with time you would develop some kind of an immune response, but there was no sensitization. It also has antimicrobial properties. Actually, it was tested for antifungal properties, and it was inhibited when you had a combination with vitamin C for some reason. It was very potent against fungi. So the regulatory status, it's approved by the FDA for food use and was given GRAS status as a flavor ingredient by FEMA. And the Council of Europe included it in a list of some substances that are not fully evaluated, but it's still approved for uses over there, as we discussed. Okay, so we had a lot of USDA funds available to us from the ARS because of the termite problem in Louisiana. So we first tested this on termites. Local trees are not resistant to these, and so 30% of the live oaks in New Orleans are infested, where the live oaks are resistant to the local termites, to the native termites, and trees in the forest are falling down. It's pretty bad. Old buildings, old churches, the beams are being eaten. And there's probably 5 billion a year in structural damage in the south of the USA alone. This bioassay is so simple, and it really works. And what we do is we chemically coat sand with the Nootkatone. We just do it by putting concentrations of the Nootkatone and the other compounds in ethanol and sand, and then we dry the ethanol, and then we put chemically coated sand on one side and control sand on the other, and you put in about 10 termites and put them in a dark place. Just close the drawer in the lab and wait 10 minutes or 15 minutes, and you pull them out. And if the termites are all on the control sand, it's a repellent at that concentration. If they're on the coated side, it's an attractant, and we did find attractants. And if they're evenly distributed, there's no effect at that dose. Such a simple assay. So we published and patented as an insect repellent for the Formosan termite. And this stuff is so persistent that if we took a popsicle stick and put 5% Nootkatone on it and left it on the lab bench for a year, and then we put it in sand and we put a control popsicle stick and we put termites in there, the termites would crawl all the way up the stick that was untreated, try to get away from the Nootkatone, and it had been sitting in the lab for a whole year. So the volatility is not a big problem with treatment of wood or other things. So we were the first lab to show Nootkatone had insect repellency. So it became a target compound for development, of course, and patent, and also the 1,10 dihydro Nootkatone and tetrahydronootkatone were targets for development. And so one day I came down the stairs in the chemistry building, and there was a poster of a graduate student who was presenting a new synthesis of vedavone. Okay, so you've all seen vedavone. That was the first compound that was related. So I went up to her office was 2 floors above mine. I went up, talked to the student. I said, "Why are you interested in vedavone?" She said, "I want to be a flavor chemist." I said, "Well, can we talk to your professor? Because we have some USDA money. We probably can support your graduate assistantship if you would be interested to work on Nootkatone instead." So we made an agreement with the professor, and we funded her up. And 2 years later, she had an 8-step synthesis. It's pretty good. So that's how we that's how we got to the synthetic part. We tested them quite a bit on fire ants. It actually kills them. We don't know why. I mean, it over time it kills termites too. We tried to examine whether it had something to do with acetyl-CoA, or we're just no idea why it affects them, but they're repelled and they die from it. So if we put 10% Nootkatone on a piece of filter paper and put it in a closed glass container with termites, 24 hours are all dead. And we can do an assay like this. We put sand in one chamber where we release the fire ants. Then we have a compound testing chamber where we put sand that's been treated, and the other end we put some food. And the fire ants, they will, if none of them are in the food side, it's a repellent. This is the way it works. Pretty simple. So here's on one side wet paper, in the middle there's sand with Nootkatones, and on the right side there's some glucose. And you can see one dead fire ant up there in the corner. I don't know why this one didn't have more, but here's a piece of paper treated with 10% tetrahydronootkatone, and after 12 hours, they were all dead. And this, just where the yellow circles are, shows you the mortality rate for the different Nootkatones. And you can see there it's higher with the tetrahydro and with the 1,10 dihydro. So if we look at the amount of paper consumption the termites will use, it's another assay, and we just put filter paper in with some glucose on it, and then we put 10 to 200 micrograms of Nootkatone on that piece of filter paper, and then we let the termites work on it for a couple of days. Then we take it out and dry it and weigh it, and you can tell how much of the paper has actually been consumed. So you can see it's quite potent. And if you compare the three compounds here, the two compounds, Nootkatone and tetrahydronootkatone, the tetrahydro is much, much more potent to restrict feeding. So this is just another repeat of that earlier slide where I showed you that Nootkatone costs $2,500 a kilogram and so forth. And that's why we need a chemical engineering situation. I visited 4 companies in Japan: Fumakila, Japan Chemical, Earth Biochemicals, and Otsuka. And they're all interested in insect repellents, but none of them have a chemical engineering unit. They all farm things out. And I found out from Baton Rouge, where there's a lot of chemistry around here, because 25% of the chemicals in the US are synthesized between Baton Rouge and New Orleans on the Mississippi River. They're just huge chemical plants here. So there's an industry of chemical engineering. And when I requested the cost of a pilot plant, the answer was $1 million to do a scale up. And so raising that kind of money was difficult. The USDA, NSF, we tried a lot of, and companies weren't interested to put that money out because they farm things out. So they would have to just pay that contract price to have the scale up even tried. So we failed to get funding to do this. So we're pretty happy now because we got funding from the NSF, and when you fund things through the university, all the stuff is done in the chemical engineering department where there's facilities are available. So that's not a cost. So $500,000, which we got on the grant for 2 years, is about the same as $1 million in industry. So we are quite excited now that we have people interested to scale up the synthesis. Japan has a big market because all of their temples and shrines are made out of wood because of earthquakes. They're flex. There's no stone buildings. And so, and they're all full of termite damage. And so they're very interested. And also they have problems with mosquitoes and ticks and so forth. And the fire ants just got into Yokohama last year, and they're very terrified because they spread all over the south of the US, you know. So there's a big market for insect repellents in Japan. That's why I visited companies there. So our main targets are really for mosquitoes, ticks, fleas, and lice. I've tested them myself on gnats, and they also repel gnats. And here's your typical mosquito who first finds a blood vessel, which I think is amazing to start with. How do they find a blood vessel? And they start to aspirate blood, and then they finally fill their tank, and then they are capable of producing eggs after that. So here's the way we test for mosquitoes. It's called the arm in the cage method. You put a rubber glove on the hand, and you leave a part of the forearm exposed, and then you put treatments on the forearm, and you time it, and you have someone on the outside observing the number of mosquitoes that land and the number that bite. Sometimes they will land and take off again when the repellent is still active. And so there's an area of the arm that's selected for the counting and so forth. And we used to be able to sacrifice graduate students and undergrads to do these experiments, but the IACUC committee won't let us do that anymore. So the professors have to put their arms in the mosquito cage. So Professors Greg Henderson, who was my colleague on this from entomology, and Mike Pirich put their arms in the mosquito cage, and they put 5% tetrahydronootkatone and isopropanol applied to the left arm, and a commercial spray of DEET, which was 29% on the right arm. And they tested them over a 6-hour period every hour in a mosquito cage. And so it looks like this. And I'm sure you wouldn't like to do this experiment. So immediately, no mosquitoes attacked either arm. They were counted every hour, and after 6 hours, 5% tetrahydronookatone was still active. The mosquitoes landed, but immediately took off. They didn't bite. But after 6 hours, and the 29% DEET arm was attacked by mosquitoes. There's been no new insect repellent developed since DEET. It's amazing. And one of the reasons is if we find a new molecule and it works, we have to take it through incredible amounts of toxicity testing and so forth. In this case, we are so lucky to find a compound that works, and it's already on the GRAS list. So the effects of Nootkatone, tetrahydronookatone, and so forth are the closest thing to market. We need to get them down to a price where you can afford to buy it, and in like a 5% solution. So we tested ticks, some local species, and the CDC tested deer ticks. They published that. They actually patented that. We had priority, so I don't know why they even did that, because we can interfere with that. But it was Nootkatone itself was very effective against deer ticks. So let's say you just take a little band of cloth and you put it around your ankle that's got 5% Nootkatone on it, and you go hiking. There's no tick is going to walk up your ankle. There's another source of Nootkatone, and that is naturally extracted from grapefruit skins and orange skins, and the cost of that is $8,000 a kilo. And the reason is that some companies and people like to have something that's 100% natural. And there are alternate competition for production of Nootkatone. There's several papers out there. They're trying to make yeast mutants that would biosynthesize it by putting enzymes from the plants into the yeast to modify valencine or modify other compounds. So the results in this 2014 paper were 208 milligrams per liter. So 1,000 liters would give you 200 grams. That's just not commercially feasible. Can you imagine running a 1,000-liter fermenter and then extracting material from it like this? So that's not feasible. This was another one that was using another yeast, and this tells you what proteins that they actually put into the yeast to enhance the P450 activity to oxidize the Nootkatone. And Nootkatone production was improved by overexpressing a couple of other genes. And the production was 45.6 micrograms per liter. So let's look at that, what that would be. That would be 300 milligrams per 1,000 liters. No way. Just way too expensive to try to extract it, so forth. I mean, Nootkatone is not soluble in water. This is not an intelligent way to make Nootkatone. Alytics tried to develop this for 10 years. They sold their company to Evolva in Kentucky. Evolva was not successful at getting the activity up, getting the production up. They recently laid off workers, and they have a catalog where you can buy Nootkatone from them, but it's the same price as the rest of the industry, so they haven't done anything. And I had one of my previous postdocs went there as a mass spec person, and I have to say, I'm sorry to say that when they tried to show results of the productivity by quantitation by mass spectrometry, he gave them the numbers, and the executives changed the numbers to present to investors. I mean, he quit. They were just not honest, too bad. But that won't work either. So we started with pinene, and that costs about 50 cents a kilogram. and it's from turpentine. So here's a synthetic scheme that we have now patented. This is an improved scheme from the first one that Ann Sauer did. And what we did is we changed the oxidation steps in the first step and in this step to ozone, which made it very inexpensive. It was first done with activated alumina and so forth, but it was too costly. So we changed it, and now this is the governing patent on the process. So what's happened here is that the chemical engineers are trying to replace 18 Crown 6 in this step. For example, they're using heterogeneous catalysts of some kind, and they try to make this particular step much less expensive. And then there's this oxycope rearrangement, which they're also working on to try to make the step less expensive and higher yield. While these guys were going through the synthesis, they did an X-ray on their product, and so they were able to show that the product was actually what they synthesized. So this was the first synthesis, was published in 2009, and then we had this improved synthesis. We did a provisional in 2015, patent filed in 2016, and had PCT in 2018. Projects are formulation of Nootkatone in soaps and lotions for Lyme disease prevention. The next step in this development is to design formulations that can be used as sprays, soaps, or lotions. And the project goal is tick-borne diseases, spirochetes, and so forth. The frontline method to combat tick-borne diseases is the use of insect repellents. Initial tests at the CDC have indicated the botanical product Nootkatone is an effective repellent. And it works at ratios of 2.5 to 5% Nootkatone. Technical grade Nootkatone is effective. It's used in the flavor and fragrance industry, considered food grade. The next step, a deliverable, will be a development of natural candidate soaps, lotions, and so forth. So here's the cost of these compounds now on the market, and the processes that have been tried by using yeast have not worked. So let's say that you had 100 grams of skin lotion and you wanted to add 5% Nootkatone, so it would cost $12.50 just for the Nootkatone in that lotion. And the industry standard is that the market price of something is at 4 times the cost of manufacture. And so that would be, $50 just for the Nootkatone that's in there. That's too high, although some people might pay it. We get it down to $500 a kilo, and it would cost $250, which that would be a reasonable cost for $10 to $12 product line. If we can get it down to $250, it would be very, very available to everybody. So Carrie Dooley, Mike Benton, and I wrote a proposal to the DOD in 2022, and it was near the funding edge, but not awarded because they had a program in tick-borne diseases. And we sent it to the NSF in 2023 in the summer and funded at $500,000 for 2 years in March 2024. Okay, so the products: skin lotion, sunscreen sprays, clothing sprays. For example, bedding spray. You could have a mosquito net that's coated. The Japanese like to hang beads from their windows instead of having screens, and so they use beads that have insect repellent on them. So that's a market. Wood treatment. It could be fire ant treatment. And also, apparently, these termites will get into museums and they will eat the wood in the frames of valuable paintings, and they'll even eat the linen or the canvas that the painting is on. So protecting art is another possible use of this material. We're looking for a manufacturing and marketing partner when we get the chemical engineering worked out. And so I think if the price gets down to even 1,000, we will capture the flavor and fragrance market because they pay 2,500 now. So there's an existing market without of $50 million a year without the insect repellency. Malaria, dengue fever, chikungunya, Zika. There's so many mosquito diseases. Now we have West Nile fever in Baton Rouge, and the brother of the girl who worked out the synthesis had West Nile fever. The Lyme disease, Rocky Mountain spotted fever are the main tick-borne diseases, and DEET is the market leader. So that's all. I'm happy to answer questions.
Thank you so much for that presentation. While everyone thinks of questions, I'm going to launch our ending poll.
You want me to end the share right now?
No, you can leave it there. That's fine.
Okay.
Does anyone have any questions for Dr. Laine? Zach, looks like.
Yeah, great presentation. So when you're going through the list of products, I see a lot of it is based on personal protection,
Yeah.
in terms of insect repellency.
Yeah.
Is there any sort of formulation or application you can see for Nootkatone as like a barrier treatment, as with like current tick and mosquito control for like a PMP? Would that be something you could see Nootkatone being used for?
You mean, I mean, where would you target the material, did you say?
Around the perimeter of like a property.
Oh, yeah, okay. Yeah, that's that. So if you put a solution of Nootkatone in the soil around the property, Nootkatone is not water soluble, so it's going to stay there for a while, and it's going to prevent any migration of termites and probably fire ants toward the property. The wood treatment we've already tried, and it works very well. I mean, the termites won't touch the wood after we've treated it with, these compounds, 5%. And so it could be used for that. I mean, get the price down far enough so well, they used to use arsenic, and now the wood that you get that's called yellow wood is not worth a thing. I mean, I've replaced my deck twice in 20 years here. The first deck was redwood, and it lasted 60 years for the previous owners. But the new wood treatment they use is just not very good.
Mm-hmm.
The old arsenic was really good, but it was terrible for the environment.
Thank you.
Couple of questions in the chat here. From Jill, would you consider soaps which the CDC found effective in removing ticks?
Would I consider what? I'm sorry, can you say that again?
Would you consider the use of soaps?
Yeah. Yes. Right. I mean, you could add it to soaps because there's a residue left on your skin from soaps, right? Yeah, usually the more useful things to add it to would be lotions and skin and sunscreens. Because somebody takes a bath in the morning and maybe they use shampoo for their hair, they'd have to have some in the shampoo, which is different than the soap they use on their body and so forth. So I think applying a lotion probably the key. I go fishing a lot. Louisiana is a great place to go fishing, and one of the really bothersome insects out there is a gnat. I don't know if you've had any experience with those things, but they'll come in a swarm, and I put Nootkatone around my neck. No gnats. so it's pretty effective. And rubbing alcohol, that's grass, so you can rub that on your skin any day, right? So that's how we formulate it.
Yeah. Jill says showering or bathing with the soap-like solution could prevent all or most tick diseases from transmitting. Please do this. It is sorely needed.
Yeah. Yeah,
Uh,
I agree. And there's this Lyme's disease thing is going crazy in the Northeast, right?
yeah.
We don't worry about it too much in Louisiana but the North, even places like Minnesota and Wisconsin are having a lot of trouble now. It's a bad disease. And the nice thing is simple thing you could do is just, you could treat a pair of socks, go hiking, right? That's all you need, because they have to crawl up here and they wouldn't do it.
Yeah. Whole bunch of chat questions here. Rachel asks, thank you for this very informative talk. I'm curious, with the grass designation, when does this come to market? Will it be EPA registered product or a FIFRA exempt project? Will it have an EPA registration number?
Okay, so I don't know the answer. So as far as we know, we anybody can sell Nootkatone without any regulation today, but it's only sold for flavors and fragrances. Okay, that's the market, right? So with this new market, it's possible we'll have to get some kind of a permit from EPA and from the FDA and so forth. But my guess is, because of all not toxicity work that's already been done, it's a really minor uh, uh, concern. I'm really happy that is not a main concern.
Great. Emily asks, are there efforts to use Nootkatone as part of public health responses? Um, for example, current dengue pandemic. I see major global benefit if products were accessible to everyone, especially those who subsist on less than $1 a day.
Yeah, so there's no program or effort to do anything with it right now, and the reason is the cost. It's just too expensive to use as an insect repellent, except by people who have unlimited money. And so it's possible that we could begin to formulate some of these things in some creams and so forth, and then just see what the regulatory people say about it. But we who are in the lab have been using this stuff ourselves for some years, just to test it for various things. And the story about the lice is, my son came home from school and there was a lice outbreak in his class. So he had these in his hair and we were petrified. It would get everywhere. So of course, we went to the drugstore and we got all the shampoos and things that they have for lice, and nothing worked. These lice are totally resistant to anything commercial. So I just got I got some 5% tetrahydronootkatone, and I went home and I dabbed it over his head overnight, and the next day, no lice. That's an anecdote, but it worked. And Otsuka tested it on a flea model in mice, and so they would put some kind of lotion on the mice, and then they had a flea test, and that was successful. So there's an animal, I mean, there's a dog and cat market, you know.
To answer Jen's question, the recording will be available on our YouTube channel. I will post that in the Basecamp as soon as I have that up. So hopefully by like Friday. Dr. Laine, have you looked at the degradation of Nootkatone under outdoor conditions, the effect of UV on Nootkatone stability?
We have not. So we would have to, there's a bunch of experiments we'd have to do for that. For example, soil samples and so forth like that, right? We haven't done any of that. The only thing that we've done is to treat wood, in the lab. Oh, there were some experiments where they treated some wood and buried it they put kind of ricks of wood in the ground to attract termites, and so I think, however, nobody looked at any of the degradation products or anything. And so, it used to be easier to do those kind of experiments. Like, for example, if you had the Nootkatone that was radioactive, you could follow it through all kinds of metabolic things if something happened to it. But now, man, you have to use mass spectrometry, and you have to look for things that you can't predict,
Another question. Is there a repellent plume or radius around a treated person or object associated with this compound, or is the distance of efficacy fairly short?
We don't know the distance. So in that mosquito test, the mosquitoes were completely avoiding the treated arm. So there's some distance based on low volatility of these compounds. And later, 6 hours later, they would land, but then they would take off. So that means that there was still concentration closer to the skin that was higher. And how much was absorbed by the skin, we don't know. And so good questions, very good questions, and they point to some good experiments that we need to do.
Looks like there are a couple EPA resources in the chat. One of them, Emily or Ben, do you want to talk about these things?
I'm sorry, you were asking about EPA stuff.
Yeah, there's a couple of our working group members have put EPA resources in our meeting chat here.
Oh, yeah. Do they have any comments? That's my question.
Sure. So, hey Ryan, I just dropped a link in. I think someone was asking about registrations and whether these would be exempt from registration. So, what they're referring to are the 25B minimum risk exempt pesticides. And there is a published list on that website of the different active ingredients that are acceptable under that exemption. But then Ben found it looks like a label where this compound Nootkatone maybe is registered. So I don't know, Ben, if you have looked at that link. I haven't. I don't know if you have a comment about that.
Well, I really appreciate what you just said. Is it possible that you could go shoot me just a little couple of sentences on that on email so I can correspond with you on this? I would like to learn more.
Yeah, surely.
I can share your email address here. Give me just 1 second.
Yeah, just my name at gmail.com.
This is Ben. Can you hear me?
Yes, I can hear you, Ben.
Yeah. Just to comment on that link, CDC actually paid to get this registered, the active ingredient with EPA, and I'm a little dusty on this, but it seems like since it was biosynthesized, it's not the same as looking at it the directly isolated in terms of the 25B exemption. But whatever the case, the active ingredient is registered, and so
yeah, these things that are
what that
natural
would mean is that any other formulations would need.
formulations could be patented. That's true. but the fact is that we had broad patent applications on the effect on arthropods, and we tested them on ticks and so forth. So just going out and doing a different species of tick is not exactly something that the patent people would appreciate as a new discovery. And but we, you know, whoever were the examiners didn't bring it up apparently because it got passed as a patent on Nootkatone for deer ticks. But and we have not challenged that for a number of reasons, because it costs money to do it with lawyers, and we don't have a market that we're losing right now for anything. So we'll just see what happens. And I'm glad they did some work on it. Let me put it that way. I mean, it gives it more credence if the CDC does something. So it's not totally harmful. But the fact that these compounds are made by biosynthesis, or they're naturally extracted, or they are synthesized like by Aramore, where they start with the major part of the compound, just oxidize it, or they do it like we do, where we start with pinene and we have 8 steps. When you get to the end, the molecule is 100% identical, completely identical. So the idea of something having some spirit because it's natural is probably, you know, anyway, that's my answer, is that all these compounds, no matter how they're made, are the same structure.
Yeah, thank you, Ben. There is a conversation about grapefruit essential oil in the chat right now. Wondering if it has enough of the chemical to be effective, and Tara says that that it's not, unless you have really concentrated and very expensive oil that wears off in a short period of time. And suggests that if you want to avoid DEET, you can use anything with an EPA label and it works. On ticks, so oil of lemon, eucalyptus with a product number, and picaridin are great options. Wondering if you have any comments on that, Dr. Laine.
Yeah, so the extracts of the orange peelings of the citrus peelings in general have some Nootkatone. They have much smaller amounts of these other 2 compounds, but they have Nootkatone. It's pretty low. And so you saw that the price of it from extracted from the rinds was $8,000 a kilo. So that's a very high price. So if there were any of it in grapefruit oil to a substantial amount, it would be much more valuable than the cost of that oil. So I think it's a low percentage. But my guess, there's probably enough of it in a grapefruit peeling. So if you just kind of roughed it up with a grater and rubbed it on your skin, you'd probably get some repellency.
Thank you. Jill says it's the tick that you don't see that transmits pathogens: Lyme, Babesia, anaplasmosis, Rocky Mountain spotted fever, and many more are fatal. And we need something that causes ticks to die off so they do not transmit them. Global warming is causing increases in ticks and pathogens. And Ben says his comment isn't about patents, but about EPA registration. Once the AI is registered, it makes it much easier to get additional products registered for use,
Oh, thank you.
or additional formulations registered for use.
Yeah.
And then he shared the product label.
So we don't know about killing the ticks, and I have to look back at the data from the CDC to see if they actually treated ticks for a long time, and we just recorded repellency. But so I don't know about the toxicity of ticks, but boy, it's toxic to fire ants and termites.
We have maybe one more question, and then we'll be about time to wrap up here.
Anyway, if you guys have my email address and you want to ask any questions or communicate with me about this project, I'm really happy to get advice or whatever your interests are. And we're also looking to try to find a company that would be interested in a market for insect repellents that might be willing to license our technology once we get the chemical engineering to a point where the scale up is feasible.
I did share Dr. Laine's email a couple times in the chat. So please go grab that if you have additional questions. Otherwise, I want to thank you all for your participation today. I think this is the most questions we've had in a while. We've had some great conversation today, so I really appreciate that. Dr. Laine, it was a wonderful presentation. Thank you so much for being here.
No problem.
And we will see you all at the Tick Academy in October, as well as our next webinar in September. So thank you all very much. We'll see you in a couple months.
Okay. Thank you.
Thank you.
Yeah, thanks for